Well, thank you very much indeed. I really appreciate being invited and coming to the Netherlands again. And I apologize, although I come to the Netherlands a lot and I haven't got a bad throat, I really can't manage your language, and I apologize for that. I hope those of you who want to have it translated will be able to follow what I'm going to say. It is indeed an honor to be here. I consider the Netherlands the most advanced country when it comes to the study of metabolic diseases. And you get things well organized, and Hanke, I think, does a magnificent job with her colleagues in bringing you all together in this extraordinary way. And I'm going to talk about that perhaps after the lecture. I was asked to give a historical perspective on the 100 years since Archie Garrod, as we call him, discovered or described formally the inborn errors of metabolism, the things that we talk about. This is a Hebrew prayer. I'm not Jewish, but it is a rather fine prayer. Blessed be the Lord God who introduces variety amongst thy creatures. It's about variety, genetic variation, that we're really talking about. But of course, when that variation is mismatched to the environment in a way, then we call that, don't we, a disease, a condition. There's Archie Garrod when he was young. He is a great hero of mine, Archie Garrod. He was incredibly bright. He was very imaginative, but he was a terrible geneticist. And he was a very bad teacher. And he spent most of his time in the back room, not being terribly attentive to the parents, but to the patients, but studying the chemistry ethically of their urine. You still don't have to get permission to take a urine sample and study it. And the condition that he studied was extremely rare, and the material that appeared in the urine, which almost had a Delphic quality in ancient times, in about 1 in a million people, was this extraordinary black material, alkapton, which was the oxidized product of a tyrosine metabolite, a protein constituent, was turned to this because of an enzyme deficiency. He worked that out. He worked out by feeding experiments, by careful studies, that it was an inability to break the benzene ring in the tyrosine molecule that led to the formation of this material, and it's spilling over in the urine. He thought it was a harmless condition. He recognized later that patients with the condition actually have bad joints, but we now know from natural history studies that actually premature cardiac disease occurs and other problems. So it is not perhaps as benign, as individual, a chemical curiosity as he thought. Perhaps he should have studied his patients rather than their urine more carefully. Well, alkaptonuria. Then there's this interesting thing that is still pretty benign. That's a 5-carbon sugar that appears in the urine and puzzles the nurses. This is another material that appears in the urine. This is his other inborn error that he first described, albinism, which I'll come to in a moment. People who just can't make very much or cannot make melanin to pigment themselves in the skin. And later he discovered porphyria. I work on porphyria. It's a dramatic illness, either causing cutaneous skin manifestations with sensitivity to light, or sometimes dramatic episodes of abdominal pain and paralysis. Often the diagnosis isn't made, and clever metabolic doctors can come along and make the diagnosis when the patient has been neglected. And you can get them better now. So this material appears in the urine if you stand it, as with alkaptonuria, in light overnight. You can test for it in the urine. And some patients with erythropoietic protoporphyria, Hanke, one you couldn't pronounce, they may get extraordinary material that's fluorescent in the plasma, and they develop photosensitivity reactions. And here is a patient who claims descent from the House of Hanover, meeting a well-known personality in Britain there, now 60 years old. She was a bit older at the time. And here are the skin eruptions, and here she is in her kitchen, very disabled by an attack of acute porphyria. So this is the original description of the inborn errors of metabolism in June 100 years ago by Archibald Garrod, a young man who was passed over for appointment for many years in his hospital, probably because he didn't teach, and probably because his ward rounds only lasted a very short period of time. So he had his faults. In his paper, he recognized that of the 16 families that he first assembled from the literature and he studied, the parents were cousins in about 9 of them. And that 12 of 36 families that he could amass and examine were affected, and 24 of the children weren't, out of a total of 36. The 3-to-1 Mendelian ratio, he didn't work that out, that came out later. So he makes pompous, long statements here about the inheritance, but in fact, it was another person who had prompted the idea of the genetic basis for these diseases. We've seen this picture before. There's the young Mendel when he was doing his experiments on plants. He wasn't just a befuddled monk with specs doing naughty things to pea plants in the garden of the monastery when he should have been at prayer. He was actually a genius, and he belonged to a community of geniuses in Brno, in now Moravia, Czech Republic. There are 7 characteristics. There are 7 chromosomes on the pea, and he perfectly described the inheritance of characters in 1865, and his work was largely forgotten about. It was Hugo de Vries in your country, a man who rediscovered the paper by Mendel in the early 1900s. And he was in communication with a British exponent of genetics, the great advocate of Mendel in the English-speaking world, who was in fact the underpinning genius of this whole area for Garrod. And I think I'm going to tell you about him. His name was Bateson. He worked in Cambridge, and he was Mendel's advocate. He was a bit of a rogue as well. Again, he had what we call in England a walrus moustache. If you were a walrus, you probably call it a Garrod moustache or a Bateson moustache. But anyway, there he is. He is a man of very strong opinions, some of them very liberal. He encouraged many women to join his laboratory, and he behaved very respectfully with them. And many women scientists grew up and became educated and published independently under his wing in Cambridge. And he was the first person to coin the term genetics, and even earlier than that date there. He was, in fact, a correspondent there, you see, of Hugo. I can't say it properly, but there's a garden in Amsterdam, which I've been to, where de Vries was. So good links between our countries. Well, what were the early ideas about gene interactions in patients and the interactions of mutant or variant genes and conditions? Well, as I've told you, we do know that joint wear and diet would influence the expression of the first one, alkaptonuria, homogentic acid urea. We do know that urinary infection and dehydration will affect the outcome for patients who have kidney stones that don't easily dissolve in acid urine, cystinuria. We know that light exposure is bad for blondes when it's very intense and they get skin cancers. And we know for people with congenital porphyria that they're probably better off living in caves. And indeed, if there was no light, this is what Garrod said himself, they could live like troglodytes, cave dwellers, and be protected from the effects of the porphyria. Otherwise, they'd be all right. He also worked later in life, did Garrod, on a much more subtle aspect of what we call the tendency to disease, the chemical individuality and the individuality of man. And he, in his later book, published towards the end of his life, Inborn Factors of Disease, described other conditions like the response to vaccination or immunization with diphtheria as being variable, as being genetically based. And he came to the idea in a very thoughtful essay, which is that one there, that indeed genetic influences made us what we are and were the result of a very long history, as we heard in the first lecture, of evolution of man. And that our variation, our carrying a certain variance at various of the 20,000 or more loci, made us what we are, but gave us a tendency, a diathesis to disease. And that was a very penetrating belief. This is the famous book that he wrote in the 1930s. And that idea was only just been taken up in the last 20 or 30 years in its full weight. He was, in fact, a bit of a prophet in that sense. Now, one of the competitors of Bateson and Garrod, Bateson and Garrod became friends because Bateson suggested it was a Mendelian factor, a Mendelian recessive gene that was responsible for the alkaptonuria. And Bateson was very clever, as I'll come into that. Bateson actually discovered genetic linkage, but it was taken to a very high level by the great geneticist Thomas Hunt Morgan in the United States, who got the Nobel Prize for recognizing that genes were on chromosomes and to understand genetic recombination, by which genes move around chromosomes and become exchanged at the time of the formation of the zygote, of the gametes and zygote. He was very rude about medics. He first of all pointed out that man, compared with his fruit flies that he worked on, was a poor breeder, he says. Hence, many families' pedigrees are too meager, too insufficient to furnish material for genetic analysis. And then the way doctors talked about tendencies to disease, they talked the cerebral type, the respiratory type, the digestive and muscular type. These are almost like Galen's ideas, the Galenic idea we heard about. Doctors that talk about asthenics, tall, thin people, and little short ones getting fat, like me. These, they thought, gave rise to certain tendencies to disease, and he just really couldn't take it seriously. Well, of course, as we know, and before Mendel's time, after Mendel's time, and really poor Garrod didn't understand this, genes are in the genome, of course, and the genome is the bunch of chromosomes that we've had described for us today, set out in color there. And on those, of course, it's DNA that was discovered in 1953, its structure, and that copying mechanism that was possible because of its capacity to reproduce. And DNA, everyone talks about it as the blueprint for life. It plainly isn't only the blueprint for life, but this advertisement for a perfume that you could get from a British store here, would you believe, was described, DNA, it's the reason you have your father's eyes, your mother's smile, and Bijan's perfume. So when we realize that DNA and genes are on chromosomes, there was a capacity to map those chromosomes to find out for a particular condition where the gene locus was, to understand the mutation and get the basis to do something with it, to reduce to the smallest particle of science that you could. And this involved fishing for genes, initially by linkage studies, which were in fact done in human pedigrees. Morgan said we wouldn't, but they were done over a long period of time. I was involved in such studies myself. They're incredibly hard work, involving collecting lots of families and studying them. I'm sure many of you in this audience will have done that. And fishing for the disease locus. So this is for quiet revolutionaries, no fishing, Her Majesty's Royal Parks there, and there is somebody going ahead and doing it. And we now know that there are numerous genetic loci based on each of the human chromosomes, even the scruffy little Y chromosome that makes us blokes the little scruffy people we are. So these are the loci that have been mapped to each of the human chromosomes here, as characterized by another great man, a great librarian, I would say, of the human genome, Victor McKusick, who died this year. He is the man who built up a computerized record of every single disease locus, every single human gene that was known in Mendelian inheritance in man, now known as OMIM, online Mendelian inheritance in man, and which you can access free through the National Institutes of Health, and enables you to get as much information as you like about a locus, where it is, what the mutations are that have been described in a particular disease. And there are now, for the diseases, 2,100 single gene disorders as of the 15th of November when I made this slide in the morbid anatomical map. So when you went to medical school to do your anatomy, that was one thing. This is the morbid anatomy of the human genome. It's not the function. It's not the molecular physiology. Well, we all understand, of course, that it's DNA that provides the substrate for variation and is really the true link between Darwin, who also had a very rudimentary knowledge of genetics, unfortunately, and Mendel, who was a genius. A copy of the Origin of the Species, annotated in its second German edition, is in the library at Brno, the Augustinian monastery there, with annotations in the margin by the abbot, Gregor Mendel. It's fascinating. He realized, deep down probably, what was going on. And we always think of disease as a disadvantage, but it's worth knowing out there's some classical examples. This is very familiar to many doctors in the audience, but I think we're a general audience. It's good to appreciate some of these things. This is a very horrible disease, sickle cell disease. It is found in 3 ethnic groups in the world, principally in sub-equatorial Western Africa, also in the Arabian Peninsula, and also in parts of South India. And it's a disease which presents in childhood, affecting the bones of the hand very characteristically, the chest causing death of lung tissue during the course of an infection, the brain causing terrible strokes in young people, and the bones which become damaged, their blood supply cut off. Initially, when you do an X-ray in a patient with this disease who's complaining of extreme pain, you find nothing, and the ignorant doctor sends them home. If he X-rays it 6 weeks later, that's what he finds, and the patient has been in agony. Because the sickle cells, because of an abnormal globin, an abnormal red pigment in the blood cells, causes them to deform, and under conditions where oxygen is limiting, to sludge, to block small capillaries, small blood vessels. The cause of this condition, the molecular cause, was identified by Vernon Ingram in 1956 in Cambridge, not 100 yards from where I work. And he was working with Crick at that time, and Kendrew and Perutz. But he published on his own, because they were very generous, nearly just over 50 years ago, because he found a peptide map, breaking up the fragments of the protein. He found a fragment that was unusual, and he analyzed it, and he showed that 1 amino acid had changed in sickle hemoglobin, and that was sufficient. And they realized, of course, that this was the basis of a molecular disease with a single gene defect affecting 1 amino acid. Extraordinary. We now know what the code for that 6th amino acid in the beta globin chain is, of course, and it's changing a glutamate to a valine. And it's just 1 base change here, as you can see. T to a G. U. Now, if you go to West Africa, you can show, as did Dr. Allison at the time first, that the frequency of heterozygotes for this trait, who are perfectly fit and well, is very much higher than expected, that homozygote children die of sickle cell anemia, but that 14% of the population here in some regions, and other regions too, are heterozygote carriers for the sickle trait. It is almost certain, in fact we now almost know for certain, that it confers resistance to the malarial parasite by mechanisms that are now being understood. It's a very defining thing. So surviving patients in Africa, individuals in Africa, are survivors of sickle cell anemia. Survivors, I beg your pardon, of malaria. We are all survivors of plague. We mustn't forget that, which wiped out 40% of the population of Europe in the 14th century. So there have been selective forces on our genome, and this is a balanced polymorphism where the heterozygotes have an advantage and are overrepresented in the population. The cost is that those who have a double dose, the recessives, they perish. That's how evolution works. It's merciless. And Darwin was obsessed with extinction, of course. Well, who would have thought that there were human mutations? Here is somebody with a form of albinism. Garrod studied albinism. It's an unusual form of albinism because this lady isn't made up. I look at ladies' eyebrows very often. But you can see here that it's not made up or plucked. These eyebrows are brown. She has blue eyes, not pink eyes like a white rabbit. But her hair is indeed, as you can see, on her head, blonde, on her arm, pigmented. And this is the equivalent of the Siamese cat and the Himalayan mouse. This is a mutation in tyrosinase, which is temperature sensitive. So if you're running about in the snow, it's very advantageous to look like this with a few leaves around, and you are protected. Because the tyrosinase that cleaves tyrosine, the same pathway as hemogenotic acid, actually, that cleaves tyrosine and turns it into another pigment, not unlike alkapton, that is the melanin pigment of the skin, doesn't work at high temperature. And so you don't get the pigment in the warm areas. But in the cold areas, like the tip of your nose, or the tip of your tail, or there, the pigment can fall. This is how things work. Well, William Bateson was the underpinning genius of Garrod, and he's undersold. I'm sorry he's English, but he did collaborate with Hugo de Vries. He was a very brilliant man. He worked out that the Mendelian factors in disease with a woman, Dr. Saunders. He worked out genetic linkage, but he didn't acknowledge that this was due to the fact that the genes were on chromosomes. That's what Morgan did. He discovered two other things. Epistasis, which is the way more than one gene may influence the same trait, and I'll come into that in a minute. One of the most powerful concepts that we now have, which he discovered from his experiments. And he also discovered homeosis or meristic variation. I'll come to that at the moment. But these are mutations of body structure and development, the so-called master genes that control the development of pretty well all animals and many plants. Well, he was an indefatigable breeder of fowl, that is to say, chickens and so on, of mice and of plants. And he lived in Grantchester, just outside Cambridge actually, by chance, in a lovely house with his name still over it. And he did the sort of experiments like this. This is how he discovered epistasis. Here are two beautiful white sweet peas. Not ordinary garden peas that poor Mendel worked on. These are beautiful, sweet-smelling peas. And when he crossed these two white varieties of peas that themselves would breed pure, all the offspring looked like this. And when he intercrossed, these parents here did a naughty thing in the middle of the night and self-pollinated those peas. This is the variety of peas that he got. Okay? He realized, did Bateson, that the synthesis of the pigment that gives the rise to these colors was controlled by genes, and that there was a precursor relationship. A went to B and B went to C. And you needed the action of one gene to allow the others to happen in full. So the genes were controlling the same pathway, controlling the same trait, which is pigmentation, but that different genes were operating to bring about the synthesis of the different chemicals that led to the formation of the mature pigment up there, the anthocyanin pigment. In a nutshell, that's what he described. This experiment actually also does show genetic linkage, but I won't talk about that. Now, we think of cystic fibrosis, the most common genetic disease that I think hospitals see in our populations in Northern Europe. And though we're very short in Britain, we're very like you. And you are the tallest people in Europe, I'm told. Seems like it anyway. But cystic fibrosis occurs at the frequency of about 1 in 2,000 live births. But cystic fibrosis can present with just sterility in a young man. It can present with a child with meconium ileus, that is to say within the blockage of the intestines within the first days of life. It can present with a bad chest. It can present with a terrible chest. And survival can vary enormously, even within the context of modern medicine. And we now recognize that many genes that influence the lung disease, that affect the capacity to combat infections in damaged lungs, that cope with the sinuses, that cope with the stomach problems, the problem in the bile, the interspissation of bile, and meconium. Many genes involved in the transport of mucus, of salts, of ions, and inflammatory indices, inflammatory mediators here, influence the outcome, the appearance of the so-called phenotype of cystic fibrosis. So epistasis, epigenetic factors are working in disease all the time, and we need to understand those because they're part of clinical medicine, part of the description, and part of the understanding of how you can ameliorate a disease. Bateson also described the words homozygote and heterozygote that we use all the time, and he was really quite a genius. Homeosis was the other thing I talked about. Meristic variation in plants. The double plants that you have in your beautiful fields in the spring, in the Netherlands, those double flowers, where Bateson realized that it was another set of sexual organs in the anthers that were turned into petals. Of course, it's the double flower in many cases. And then he realized, and these are original ones from his book, 1894, he's obsessed with understanding species, obsessed with Darwin, wanted to understand all this. He realized there were genetic tendencies that give rise to human deformities, as we call them. This is brachydactyly here, where there's a failure of joint formation in the fingers and toes and elsewhere. And then Morgan, of course, described these mutants after Muller actually described these mutants working in Morgan's lab after irradiating fruit flies. Here's one, instead of having antennae, it's got legs. Antennapedia, it's got legs on its head. And these bithorax, you can see, it's got a failure of segmentation. Well, Bateson was right. There are genes that control the formation of patterning, of the development of segments, of the development of your normal joints in your hands in brachydactyly. And here it is, again lifted from his paper there. Here's a paper by Garrod et al. A few years ago in Nature Genetics, and you can see, in fact, that they studied this condition. Here the X-ray appearances of brachydactyly, here the rather short individuals. Here's the pedigree. It's a dominant condition, and they showed a mutation in a developmental gene known as Indian hedgehog. Geneticists are so clever, they give bizarre names to things. I won't come into that now. But that gene is mutated in brachydactyly and in many other developmental abnormalities. You see these master genes which have been mutated. There it is. This is a very horrible condition, causing a great distress to parents of children born. Hosoprosencephaly. I'm sorry to show these gory pictures, but I just want to illustrate the point. This is a child with a small brain very often, with a failure of fusion across the middle, and sometimes palatal abnormalities. But by genetic study, it's been possible to show that there are 5 variants of this, all with different determinations, all genes that influence the development and control and formation of our face, fusion of our palate, development of our brain, settling in the skull, and the rest. Extraordinary. Some of these include the hedgehog gene. This is Sonic Hedgehog, which is taken from a computer game here. And some of them, this one here, is TGF signaling gene effect. And this one here affects an enzyme involved in cholesterol biosynthesis. And that means you might be able to interrogate this. Not only discover these in a prenatal diagnosis, if that's ethically acceptable for the family, but you may be able to get a handle on improving the behavior of that gene product. So understanding the absolute reduction of this, not like a historian, I'm afraid. I disagree. You have to get right down to this. It takes heroes to pursue this. So that was Bateson's contribution. We begin to understand much more. For example, segmentation in all animals involves homeobox genes, homeotic genes, whose products diffuse down the length of the tadpole or the worm or the human to involve the formation of the segments, the vertebrae, the tail. It's extraordinary. People are beginning to understand these things now. And they've been discovered through mutations, through variants. Well, through understanding genetics, it's possible now to create your own. Last year's Nobel Prize went to Martin Evans, Oliver Smithies and Mario Capecci. Martin Evans is a colleague of mine. I don't want to boast about Cambridge, but he wasn't recognized for many years fiddling around, and he described embryonic stem cells. And embryonic stem cells can be manipulated, and they were manipulated to generate mice with cystic fibrosis that don't quite imitate human cystic fibrosis, but they're very interesting, and many other genetic diseases, so that you can study the disease perfectly once you know, by linkage study, by fishing for genes, once you know what the mutation is. It's wonderful stuff, carefully regulated, to be able to generate a model of a disease precisely mimicking what the human condition is. A couple of weeks ago in Nature, this appeared, in Science, this appeared, which was doing transgenic experiments in pigs. It's possible to get an authentic model of cystic fibrosis that affects young babies with meconium ileus in pigs by following the embryonic stem cell pathway. This is remarkable, and it shows how we can begin to understand what we need to understand to get to the bottom of these diseases, so we can think about improving the lot of those that suffer from them. So, what about complex diseases? I'll just touch here very quickly. The fishing is much, much more difficult if you're looking for complex traits. I told you that cystic fibrosis was no longer simple. It's not the CFTR gene that was identified in 1989 anymore on chromosome 7. It's all those other effects that come in epigenetically and epistatically to change the phenotype in the person that you see and their susceptibility to the manifestations. But for complex traits like diabetes, like obesity, like other conditions, hypertension, inflammatory bowel disease, the lessons learned from genetics of single gene disorders are being extended. It's a very, very long haul indeed. And of course, not everything about life is going to be in the genetic blueprint, but you need that first. And mapping those is very difficult indeed. There's a very thoughtful article, in fact, last week published in Science by Eric Lander, in fact, who is a very thoughtful person. How you can do massive deep sequencing, how you can look for tiny little variations throughout the human genome and identify traits that determine diseases. And people want to do that for all sorts of diseases. We were hearing earlier, you know, the big diseases that catch all the attention that we all feel and know about. Breast cancer. We all want to know about susceptibility to breast cancer. It's difficult because you have, of course, genes which are big determinants, where there's a tenfold increase in the risk of getting breast cancer if you belong to a family where BRCA1 and BRCA2 genes are segregating. But those gene variants occur rarely in the population. And they occur perhaps 5% of patients. And they're big genes and they're difficult to sequence. Then there are rare, moderate risk disease where about a twofold chance of increasing. That's bad news. You don't want a twofold increase in the chance of getting that disease, of course. But what do you do with that information? And then there are multiple genes which are very common, genetic forms of various alleles, all these very complex variants here. This is fibroblast growth factor receptor 2, for example. There are variants in that, which to a very low level will have an effect. If you have that and a few others, well, you might need a bit of screening. How do you handle this information? How do you get to causation? And this is the big problem. How do you get to causal determinants in genes for complex diseases? And it's the lessons learned from the single gene diseases which are driving this forward, combined with advanced technology for deep sequencing, finding copy number variation and various other complex changes in our genome that make us actually the individuals we are. So I'm going to go through a historical summary, and then I'm going to talk about what we all want to talk about, I think, as best I can. We've learnt about the metabolic basis of inherited disease. As Bjorn Furling, of course, described the first treatable one by dietary measures, phenylketonuria, 1 in 14,000 approximately the European population. And we screened for that at birth. And they were in communication, those two, before Garrod died. He was thrilled to bits. Molecular disease defined by mutation analysis. I told you about Ingram and Linus Pauling, who described sickle cell anemia as the first molecular disease. Then there's been rational therapy based on understanding the enzyme deficiency, what goes wrong, the transport defect. Galactosemia, a diet, penicillamine for Wilson's disease and cystinuria, various cofactors and vitamins for beta-crotonal, methyl-crotonalurea, and all those interesting, difficult conditions. You can find factors that can help. This is very good, it's very encouraging. It's not enough for the 2,100 that you're talking about. Then there's been the molecular period. Site-directed mutagenesis, protein engineering, crystallography, transgenesis, as I talked about, generating your own models in different cells. Before and after the introduction of the polymerase chain reaction in the mid-1980s, which enabled us to do rapid reproduction, rapid synthesis, rapid analysis of mutant genes in a test tube. Previously it was take a whole lab years to be able to get to one gene. That just quickly accelerates your process of isolating, purifying the DNA and characterizing mutations, and enables you to do big sequencing projects. And then the Human Genome Project, leading on to the genetics of complex diseases. So we're in the genomic phase, and I get bored with genomics and economics and endless reviews about these things. It's all about economics, mainly for that lab. What I'm interested in, which I think is what you're interested in, and I'd like to think that Bateson and possibly Garrod would really be interested in, is the application of molecular therapy, specific treatments based on understanding. What's in it for us? The genetics for the human race, the medicine of inborn errors of metabolism. This is a bit of a quip, but it is true that cats don't ask for medicine. In fact, you have to ram it down their throat. But humans do have a desire to take medicine. Certainly when I get migraine, I like to be left alone. And so the desire to take medicine is perhaps the greatest feature which distinguishes man from the animals. That's very cynical. But I would say that healing is not just a matter of time. It is indeed a matter of opportunity. And the opportunities come from understanding, not just empirical, but deep understanding. And here is an example. This gentleman, a Japanese researcher, very dedicated in the, I suppose, on the hinterland, the borderland of academe and the pharmaceutical industry. Akira Endo got a Lasker Award this year, 100 years after Garrod. And he got it for the discovery of the statin drugs. He was working on an exceptionally rare inborn error of metabolism. The work having been done by two Nobel Prize winners, Goldstein and Brown, who described mutations in the receptor for taking up cholesterol and circulating it into cells for uptake. And when it's defective, one mutation, you have a very high cholesterol. If there are two mutations, homozygous familial hypercholesterolemia, then you have very high cholesterol, terrible vascular disease, heart attacks by the time you're 12 or 15. And we have patients with this, the treatment for which tends to be liver replacement therapy. He worked on this condition. He worked on finding an inhibitor of the pathway for the biosynthesis of cholesterol. And he found one. From this mold here, Penicillium citrininum. And he extracted from those the first inhibitor, compactin, which is a statin. And it blocks the biosynthesis of the first committed precursor of cholesterol, mevalonate, mevalonic acid, and thereby can reduce the cholesterol concentration so that this patient was able to have a child and not die of a heart attack. Dr. Yamamoto's patient. It was all rational. 2 Nobel Prizes for understanding the biochemistry, 2 Nobel Prizes for understanding the biosynthesis, the uptake of cholesterol, and then a great deal of work searching for an inhibitor for that pathway. Rational drug design. And of course, the prescriptions for statins are billions and billions of pounds and dollars, and somebody thinks we should all be taking them. Certainly, there are very large numbers of them used for the common diseases that lift the cholesterol concentrations and predispose to heart disease and stroke and peripheral vascular disease in the population. Again, a very big lesson learnt from a rare disease. Well, we're talking about orphan diseases, aren't we? And I know that Hanke and others have been very important in driving forward work to ensure that orphan diseases get recognition. They're the ultra-orphan diseases too, where perhaps there's 1,000 or less individuals in your country who are affected. The lysosomal diseases that I work on, which you've heard about, of which there are 70 in fact, occur with a birth frequency of 1 in 7,700 births, but they individually are extremely rare. In the UK, the most frequent one, Gaucher disease, affects about 300 patients in the whole entire country, and MPS 1 less than 100 patients in fact. The lysosomal diseases have many manifestations. Some affect the skeleton, as does Gaucher disease here, infarcting bone as with sickle cell anemia, leading to thinning of bone and collapse, and large masses in bone causing great pain at the end of long bones. Affecting the nerves in metachromatic leukodystrophy, the brain here, as in Sandhoff disease, in Tay-Sachs disease, the nerve cells around the retina, and this poor child here, as shown with the condition. The muscle in Pompe disease, described from your great country. This little boy with GM1 gangliosidosis. This little child with type 2, type 3 Gaucher disease with a large liver and spleen, very anemic. This type 2 Gaucher patient. This little girl with sulfatide deficiency. And this little boy with MPS 2, as shown here. This is multiple sulfatase deficiency, and this is Daynon's disease. They're just a few affecting the heart. There's an opportunity with lysosomal diseases recognized from the outset by their discoverer, Christian de Duve, who was Belgian, Low Countries, born in Thames-Ditton. Brilliant scientist, got the Nobel Prize in 1974 for describing lysosomes and peroxisomes, but he recognized from the outset that the lysosome would be accessible to medication, to proteins, to drugs given outside. And you could correct, if there were abnormalities, which his colleagues later found, you could correct those abnormalities by feeding something, by providing something. The very simple concept, as with diabetes, if something's missing, just give it insulin for type 1 diabetes. So if you could get the address right, the receptor that was responsible for uptake, you could in fact access that compartment and correct an abnormality. The most dramatic commercial example of this has been the correction of Gaucher disease with work from the NIH, amalgamation with the National Gaucher Foundation in the 1980s, finding $3 million to launch a trial of an extract of crude missing enzyme, glucocerebrosidase, with targeted delivery and address to the lysosomes in the macrophages that are affected, an amalgamation of industry driven by patients and academia. Ceridase. Hundreds of tons of human placentae went to the formation of ceridase, but I'm old enough to remember its first use and used it in the first patient with Gaucher disease to be treated in the United Kingdom in 1991. Now licensed, this one, the recombinant version through genetic engineering, as Cerazyme, a perfectly pure version. There have been fortunately no complexities, no complications of using tons of human placentae to produce ceridase, which is now not in use much. And this is some studies we did just that shows the targeting of those preparations. We radio-labeled them and injected it into patients at certain doses, and a healthy controlled individual here. And we're able to show targeting to the sites of injury in the liver and spleen. Is a massive spleen that had to come out for that reason there, of course, unfortunately, with this disease, 4.5 kilos worth. But you can see the targeting of the protein to the sites of disease, here at the ends of the long bones, and here to a massive liver and to the shoulders. It does work, and it does correct the abnormalities of this disease. Previously, with palliative surgery, taking the spleen out, bone marrow transplantation, all either risky or not definitive. But here's the first patient in the UK to be treated. He was anemic. He had a low platelet count and required transfusions for bleeding. He was wheelchair bound because of bone disease, as shown by MRI scans here. And within 3 weeks of treatment. Here, of moderate dose of the treatment, his blood count's corrected. He's been maintained on quite a low dose ever since. That's 1991. He got re-employed. He's walking about. He's perfectly healthy. He's never had another transfusion. In fact, I've taken lots of blood from him since. Astonishing. And so it has been for many thousands of patients treated for this condition. Nothing succeeds like success. And money can't buy you friends, but it buys you a better class of enemy. And these companies here, including this home delivery company, are in competition. Would you believe that for the small numbers of patients with Gaucher disease worldwide, an ultra-orphan disease, that there are 5 at least companies, there are others, battling in there to get a piece of the action? Yes, there are. The incentives through the Orphan Drug Act have worked. Here's a remarkable case where another company, in conjunction with the Genzyme company, BioMarin, produced aldurazyme, a recombinant iduronidase, which is used for MPS I. And this young girl was very crippled. She gave a classical story of a genetic disease, presenting at the age of 4 with umbilical hernia, age 7 with tingling in her hands and stiff wrists which had to be operated on, the usual palliative surgery, invasive, painful surgery. She had a cardiac murmur. She was so arthritic she described herself as being like a granny. The diagnosis was made when she was 10, a 6-year gap. She had 17 procedures for fixed joint deformities, wheelchair bound and all the rest of it. Within 1 year, she was able to do her hair, write essays, walk to classes where she studied to be a student radiographer, that photographed there in the national newspaper in her room in Exeter University in the UK. A lovely young woman gives a very powerful account of the idea of utility, progress, commitment. And these treatments are very expensive. If there were 10,000 patients with MPS II, the treatment would be cheaper per person. Of course, the drugs get a certain levy, and we understand that. But these things really are about what society wants to do. Enzyme replacement therapy, targeted therapies using various addresses, I won't go into the details now, have been licensed for all these treatments, all these diseases now. And others are coming along stream. Pompe disease, a dramatic effect. Even some of those that affect the nervous system, we're not quite sure how they're going to work. What are the specific treatments for lysosomal disease? They're a good model. Well, marrow transplantation was one method and is still used for some indications, but it's pretty dangerous. Enzyme replacement therapy, a method like a statin to reduce the synthesis of the substrate. There are chemicals, amino sugars and other chemicals, biochemicals, as small molecule drugs, which will enhance, will reduce the amount of build-up of the material, like they reduce the plasma cholesterol by reducing the action of the biosynthetic pathway at the level of mevalonate with the statins. They're kind of statins for this area. Enzyme enhancement, stabilizing the mutant gene that doesn't quite work and doesn't last long, like those tyrosinase mutants. Gene transfer and stem cell therapies, they will come. They will definitely come. It is now possible through the work of Dr. Yamanaka and colleagues to take cells in a biopsy, skin cells, and culture them from a simple biopsy in the skin of an animal or a human, and it's possible to then render them pluripotential. They could become any cell. They could populate any organ of the body. They could even go germline, as shown here in this animal. It is possible to change them now in a way that does not involve integration into the genome safely, in a transient expression system that enables them to change. Well, even in the lysosomal diseases, there are those that are almost beyond reach at the present time. Of the 70, many do affect the brain and the nervous system, and these are very difficult to bear. So even amongst the family of lysosomal diseases, there are those where there is a treatment and pharmaceutical investment, and there are those which is pending. And Tay-Sachs disease is a very rare disease, fortunately, but it's an iconic disease of this kind, emblematic of the serious diseases where a child is born normal and healthy, but progresses rapidly in a horrible way with blindness, psychomotor retardation, enlargement of the head, motor deficits in a rapid period of time, with atrophy of the brain and the cerebellum, with the disease you've seen before. And this young girl here died only 2 weeks ago, aged 6 years and 9 months, the oldest survivor from that condition ever, and she had been taking a substrate-reducing drug. Well, I wanted to end on a good note and just something of our own. I hope you'll forgive me, and I've just got a few moments to show you. This is a model generated by transgenesis of those diseases, Tay-Sachs disease, Sandhoff disease, similar, in the mouse. This poor little mouse lives to 16 weeks. Like all mice, it is in fact doubly incontinent. But you can see that it's in a desperate state. It's trembling, it can't feed for itself, and we have to kill them just a few days after that. They die 16 to 17 weeks. They were generated in order for study of these conditions. My colleague, Dr. Begonia Cashon, has worked with me for 11 years on a gene therapy project, which everyone said was mad, has stayed with me, and we found the funding somehow to develop gene therapy for this using a particular system that she designed, and my gosh, we chose well. The problem with this condition leads to an enzyme deficiency, and this pink staining here is a section of the brain of a mouse, which is a normal little mouse running about at about 35 weeks of age. In the deficient animal, the brain is white. There is no activity at all. Giving one injection of her therapy in one side of the brain, in fact the right side of the brain, into the striatum here, on one occasion, the animal recovers very quickly after this. Operative mortality less than 1%. After 34 weeks, you can see that the entire half of that brain and half of the spinal cord, which is laid out here for you, went red. In other words, there was abundant activity 34 weeks later. So what I want to show you is that animal killed at 35 weeks of age is rather affecting, and I'm sorry it's rather horrible, but don't forget that the injection was on the right side, and there's a crossover. So we would expect the left side to be corrected. And can you see? He's got a normal left movement and left. It's rather affecting, I'm sorry, but you can see that the left side is moving normally because it's corrected transcursively across. Yes? Okay. Now, with 4 injections, these animals live forever. This is our first old animal. He's 2 years old. He's got a bit of a hump. But he's running about perfectly normally. He got out one weekend and did things which were not allowed by the Home Office inspector to the lady mice in the next door cage. We're never quite past it, us chaps. He's able to smell the ladies, and I don't know what time they had, but he had a great time. He's not quite normal, but he's 2 years old. And when we took his brain and looked at it, the entire neuraxis of spinal cord and brain, this is a normal, this is the knockout transgenic mouse, white. There's the whole section of the brain put on the slide. And here, throughout the entire neuraxis, 2 years later, after a single treatment, the entire brain is corrected. And the storage material that builds up in there is gone, the inflammation has gone. Well, we need to translate to the clinic a bigger brain. Mice, you've heard about mice. You need a bigger animal, a more complex brain. Something like that, that's almost rivaling ours. And this is a spontaneous model of the disease, very affecting. I love cats. In a 12-week-old cat with Sandhoff disease. This was in Alabama, where we do these experiments with Doug Martin, Nancy Cox, and Henry Baker. This poor little animal really can't cope. Cerebellar disease, weakness of the legs, and the other little competitor would normally take a swipe. Even they feel sympathetic. This is an animal with a similar disease, feline gangliosidosis, 7.5 months after gene therapy. This was done by our colleagues again, and we collaborate with them on this. This is a very big brain, the cat brain. It's complex. This is remarkable. I'm nearly done. So the needs, as I see it, we need to stay together. We need to plan for people, doctors and nurses, and Hanke, even if I may say so, your successors, to work. Can't just leave it up to the charismatic megafauna, the big stars. We need people to all sign up and work in this area, in the laboratory, in the clinic, to describe these diseases. There's no question that united we stand, disunited we fall, all of us, there's no question. There must be continued incentives for the pharmaceutical industry. It's a mark of Western countries and developed civilizations that they believe in these things enough to have pharmaceutical companies. I don't have any shares, to make the world a better place. That's why people do this work. There's not been a single drug, to my knowledge, developed in the Soviet bloc, post-Soviet bloc. I don't want to be rude, because it isn't just kind of humane to do that. Take other people's maybe a little bit. They're the old medications of China. But the concept of making drugs to improve the lot of your society and to pay for it and to sign up to it is a societal thing of the West and is something to be proud of and celebrated. But there need to be continued incentives so that specific therapy for individual patients and the early detection of disease and ethical prevention of the disease in an acceptable way in the population. I've never shown this slide before. That little boy had cystic fibrosis. He was 18 at the time of this picture in a boarding school in England many, many years ago. That boy, he died, by the way, 2 months later of the complications of his illness and was teased throughout his time at school. This boy had Marfan syndrome. He died at 34 years of age. He fathered 4 children, all of whom I have seen. They have, some of the 3 of them have Marfan's disease, a ruptured aorta. There is treatment for both those diseases today, and more is online. The sartan drugs work for this condition. I'm pleased to say that one of his children is taking them. And I had to watch this, and I thought it was terribly bad, and I don't want this to go on anymore. So I think that we all should be signed up to research and patient power and pharmaceutical investment to do what society really wants to do. Here's old Garrod at the end of his life, and what he said was very moving. Obviously, clinical medicine presents immense fields for scientific research. This is his lovely essay, The Debt of Science to Medicine, What We Learn. And those who cultivate them have the added satisfaction of knowing that every advance of medical science will, sooner or later, bring in its train some forward movement of the healing art. And Einstein also got it right. I never think of the future. It comes soon enough. Thank you very much.